Good afternoon, everyone. Thanks for joining us. We're here for an amazing webinar about medicines from the sea. So I'm Jack Barth. I'm the Executive Director of the Marine Studies Initiative, and we're happy to welcome you. We're co-sponsoring this event with the College of Pharmacy. And with no further ado, I'd like to invite Carrie McPhail. She'll be hosting this wonderful webinar. Carrie is a professor of natural products and medicinal chemistry in the College of Pharmacy's Department of Pharmaceutical Sciences. Her research is focused on the discovery of new natural products from diverse marine habitats. They have potentially new biological actions relevant to cancer and infectious disease research. I just want to do a shout out to Carrie. She's been with us in the Marine Studies Initiative from the very beginning as we planned to pull together the natural, social sciences, arts, and humanities around a marine theme. And so we're looking forward to the webinar, and I'll introduce you to Carrie McPhail.
Thank you, Jack, and welcome all wherever you may be joining from. After some introductions, we will have our keynote lecture followed by a discussion panel starting around 10 minutes after the hour with OSU faculty who will describe their marine natural products research before a general question and answer session, and of course the closing comments. Just since mics and cameras are disabled, we encourage you to ask questions throughout the webinar via the Q&A function. Please type in questions at any time and we'll answer as many as possible during the Q&A. The lecture today is being recorded and will be posted on the Marine Studies Initiative YouTube channel. So today it is just my great pleasure to host Dr. Barry O'Keefe as Chief of the Natural Products Branch of the Developmental Therapeutics Program at the National Cancer Institute. Also with us are three of my OSU pharmacy colleagues. Dr. Jane Ishmael is Associate Professor of Pharmacology. Dr. BJ Philmus is Associate Professor of Medicinal Chemistry. And Dr. Taifo Mahmud is Professor of Medicinal Chemistry. Helping us also with the audience Q&A session is Hayleigh Middleton, who is a third-year biology pre-med undergraduate student here at Oregon State University. And Hayleigh is also the field trip coordinator for the MSI Ocean 11 Club. So to introduce our keynote speaker, Barry O'Keefe wears several hats at the NCI. He serves as director of the Molecular Targets Program and head of the Protein Chemistry and Molecular Biology section, as well as being chief of the Natural Products Branch. So for this career path, he earned a Bachelor of Science degree in botany from Michigan State University and a PhD in pharmacognosy from the University of Illinois at Chicago. So then in 1994, he joined the NCI Laboratory of Drug Discovery and Development to study novel proteins from natural product extracts. And now, as Chief of the Natural Products Branch, I invite Barry to speak about his program's ongoing campaign to test natural product extracts from plants and animals collected around the world and to develop new medicines. Thank you, Barry.
Thank you, Carrie. Thank you very much. Here, I'll go ahead and share the screen quickly. I wanted to say first off to everybody out there, it is my pleasure and my thanks to Jack and Carrie for inviting me here today. A couple things just in general, since this is targeted towards you undergrad folk, is that, I never thought when I started as a botanist back in the day that I would end up working predominantly with marine organisms later on, but you never know where to go. I think you kind of just have to chase your joy. Throughout my career, I think people have always told me that my choices were poor and that there were lack of job opportunities in them. But fortunately, they proved to be wrong. And botany and pharmacognosy turned out to be excellent areas, and researching proteins from marine organisms did as well. So stick with it. Don't get disheartened just because people tell you that in your careers. Anyways, what I want to talk to you about today a little bit, just to give you an outline, is natural products as drugs in general. Then we'll talk a little bit more directly about marine natural products that have become drugs. Then we're going to talk more generally about natural product-based drug discovery. And then a new big program here we have at the NCI, which is trying to facilitate making that easier for everybody. Then we'll wrap up with a little bit of biotherapeutics from natural sources. And the goal of this at the end is that you're going to want to do this because this stuff is cool. You get to dive in cool places. You can see some of the pictures here from the collections from the NCI. I will let you know we have over 25,000 underwater photographs of marine organisms that were collected for the NCI over the years. And between the beauty that you get to work in and the fact that pretty much every piece of equipment we work with costs more than my house, it's kind of cool. So let's move along. You, as undergraduates, might be familiar with several of these compounds: penicillin, aspirin. These are all commonly used drugs. If you're a pre-med, you've probably seen a lot of these compounds listed. What you might not have seen, however, is the sources of all of these. And these are the source organisms for all those compounds, which are part of what everyday doctoring in the United States and the rest of the world is using these all the time on a daily basis. And they all came from these source organisms. So there's a big contribution of natural products to drugs in the pharmacopoeia everywhere in the world. What you might not be aware of, unless you've been watching some of the slides as they go by while we were warming up here, is that marine organisms are also making a big impact in drug discovery. So you see, these are several marine organisms here, a wide variety of looks to them, and they've produced a wide variety of molecules as well, a broad chemical diversity produced by these organisms. And importantly, all of these compounds then have been turned into drugs that are used for a variety of reasons. Some, very importantly for us at the National Cancer Institute, to help, knock down cancer and give people additional years of life because of these compounds. And so if you put them all together, there are thousands and thousands of people who are alive today because of the compounds that came from these marine organisms. I'm going to take one in particular and stress it in a moment here, but I thought I'd let you know, like everything, there is a website that you can go to and find information about all the drugs from the sea. And here there's currently over 800 clinical trials ongoing with marine agents for cancer and other diseases. There are a bunch of additional drugs that are currently in phase 3, 2, and 1 trials. And one of the biggest areas that marine natural products are making an impact in drugs is with antibody drug conjugates. I'll talk about those in a little more detail here. So on this image, what you see in blue is a depiction of a monoclonal antibody. You might have heard about these in your classes, and they're used commonly with drugs today. What you see here in green are warheads that are attached to these antibodies, and these are toxic molecules that then use the antibody to target a cancer cell, and then the toxic molecule results in the death of that cancer cell. And one of the main sources for all of these small molecules that are used and attached to these antibodies is this lovely looking creature here, known colloquially as the sea hare. And this organism produced a very unusual group of molecules that you see here, dolastatin 10 being the lead molecule of that group. These were found by a group at Arizona State, that coastal university. And so they actually were allowed to go out on a boat with friends from the West Coast. So good on you for allowing them to join you. And 2 molecules came out of this, MMAE and MMAF. And these have now been attached to a variety of antibodies. There are at least 4 very important anti-cancer drugs that are made in this manner and are saving lives today. And there are probably at least 26 additional clinical trials of these type of molecules attached to antibodies for cancer treatment. So this is part of kind of a revolution that's going on with targeted cancer drug therapy currently. And marine natural products are right at the point of the spear of that. So as you might imagine, I'm kind of big on natural products. This is there's a lot of different ones, not just from marine organisms, but from all sorts of organisms. And this is just a group showing kind of the chemical diversity. The same way there's biological diversity in all of these different ecosystems, they produce chemicals that are also greatly diverse. And that's very important for drug discovery. And what we can show you is that some colleagues of mine, my predecessors in the natural products branch, routinely go through and look for drugs that are produced from natural products. And as you can see over time, this is showing all of the small molecule drugs produced that are based on natural products on a year-to-year basis. And what you can see is that over time, for the last 40 years practically, at least 25% of all small molecule drugs are natural products. And so that is something we can all be proud of in the natural product community. However, there's a problem. And that's if we look at screening, people test compounds by putting them in with cancer cells. Do they kill the cancer cells? They put them in with viruses. Can they block this virus? And what we're seeing is that though there's a huge increase in the number of screens that have been done over the years, you can see here the data from 1993 through 2017, only a very small portion of those screens actually include natural product molecules. And that's a problem. I mean, it's great that we get 25% of our drugs out of maybe 1% of the screening, but what happened if we could maybe increase the amount of screens that include natural products? Could we even get more out of that? So the NIH overall decided that natural product research is a core opportunity for the future. It has decided that for at least the next decade, this has the potential to transform the healthcare field. And so they've made uncovering new opportunities for natural products a priority at the NIH. And one of the reasons is because the NIH and here at the NCI, we have a huge repository of natural products. So here where I'm showing is a map which shows where our collections of natural products are from. If the hexagon is larger, it means it was a larger collection. They're color coded. So those in blue are marine organisms, those in green plants, and those in yellow orange here are actually microbial collections. And you can see we have almost 240,000 natural products, of which marine organisms make up 20,000. And I will let you know, we just got some more of those. So that's up to 25,000 now. So if we get about 40% of all clinically used anti-cancer compounds come from natural products, but less than 1% of the high throughput screening, our problem here at the NCI is how to figure out to increase that use. And so there are challenges to natural product work with screening, time and cost of isolation, resupply. These are challenges, but as shown here in the recent cover of the Journal of Organic Chemistry, nature is a much better chemist than we humans have been able to replicate over the years. So they can make a diversity of molecules that we can't find anywhere else. And that amount of chemical diversity is vital to us finding new drugs and other agents for the future. So what are the challenges? I'm not going to go through this with a whole lot of detail, but natural products are difficult to screen in their crude form because they have a lot of compounds in there, thousands of them potentially. And so some complicate cell-based assays, some cell-free assays, and they have high hit rates. Also, each extract contains, as I mentioned, lots of different compounds at different concentrations, which makes it a bit of a challenge. And also then the purification and structure elucidation traditionally have been kind of time consuming and slow. And those methods haven't always gone well with high throughput screening in the way modern drug discovery is done. To just show you a picture worth 1,000 words, this is what a plate full of crude natural product extracts looks like. They're colored, they're viscous, they're kind of sticky. They can really kind of mess up your fine equipment. And if we take a look at assays done with natural products, if you take a look here and just ran an assay as done in any drug company or large screening center and didn't modify it at all and ran it with natural products, what you would see is data that kind of looks like this here in red. And what you see is this is a huge hit rate. 40% of all of these extracts showed activity in this assay. That's a very large group. But if you then modify your assay appropriately for working with natural products, and there are techniques to do these, we published on this just last year, you can reduce that hit rate to 0.3%, which is a very responsible hit rate and a good way to run the assay. So just because things like this, what you see on the left, doesn't mean you can't still get assay data like you see on the right. So what did the NCI do? We're the NCI, so we have workshops and other things like that, bring people in and decide on things, but we got two different divisions of the NCI to work together, and they created the NCI Program for Natural Products Discovery, which is going to facilitate research both in the NCI and outside of the NCI. And this is actually funded by the Beau Biden Cancer Moonshot Program. And it has a lot of specific aims. I'm not going to go into all of these in detail, but part of this is building new technologies. We have to start from the ground up here and figure out new ways to do things, put those into action, create a partially purified natural product library that'll be much easier for people to screen, and then help them isolate and identify molecules from that. And we also want to expand the chemical diversity from culturable organisms so that we don't have to go back and cut down a plant or collect something from a coral reef if we can avoid that. So some first things first, we have, as part of this program, access now over 20,000 new soil samples from a citizen science program at the University of Oklahoma. You can see on this map of the United States where these samples have come from. And so these are a culturable organism. About 12,000 of those we already have photographed vialed. We also have received recently, through an agreement with Australia, over 4,500 new marine macroorganisms from Australia and over 7,000 marine microbes that will be growing up here and extracting and making those extracts available to the public for research. So what we want to do now is take this big library of 200,000 things and turn it into something that's easier for everybody to screen. And so what the goals were of that were to create a library of over 1 million fractionated extracts. And to fractionated extract is say something that's crude like this you see here in this vial and turn it into semi-pure molecules that you see here. And so that's the process of pre-fractionation. And what the goal was to be able to support at least 20 different groups screening this for a decade, and then to have 90% of that contain enough material that we could work on it. And then everything had to be barcoded, put in tubes. We had to then plate this all out in 384 well plates and make 250,000 of those. This all had to be automated in a storage facility. Everything had to be QC'd and barcode labeled like you see here. So it's a huge undertaking. And so I will let you know up front, my job is generally to make outrageous requests for people to do things that have never been done before. And then we sit around tables and figure out how to do them. So what was our goal here? Well, basically we were taking natural products chemistry, which has been done, if you went back and looked 20 years ago, much like it had been done 100 years before that, with large columns like this, separating natural products that you see separated here in bands. But what we wanted to turn it into was something new and much more automated, that we could have a much higher throughput that would match the kind of throughput people use in drug discovery today. And to do that, we had to do a variety of things. What I'm showing you here is we had to actually make a lot of the components that we use. So we started out being a regular lab that just did natural products chemistry. We now have 3 different 3D printers to make components for robots, because we've had to help design robots as part of this to do things that they were never designed to do before, which is kind of cool. So what you see here in the upper right-hand corner is one of those robots that we worked on with the company Tecan to allow us to do 88 simultaneous pre-fractionations in an automated manner in less than 3 hours, something that normally would have taken weeks potentially for an individual lab to do. And so we reduced that to practice, and we've now produced over 450,000 fractionated natural products. And we've released the first 326,000 of these to the public. So they're available for free to everybody, everywhere in the world, to screen against anything. And so that's pretty cool. We have also, as I mentioned, have over 20,000 new soil fungi and 6,700 marine microbes. These we bring up, we photograph, we have done ITS sequencing on them so we know what they are, and we are also making those available. So far we've had greater than 60 requests from screening centers to receive this library, including, I would like to say, Oregon State University. Dr. Van Bremen has signed an MTA to receive these extracts, so you guys there can play with them if you like. I will say, in the first 2 years, even though we've had this pandemic, which has totally been unfortunate, we have shipped over 4.5 million samples from this. And to let you know how that is in the big scheme of things, that's more samples than were shipped from the natural products branch in the previous 40 years of its existence. So in the last 2 years, we've shipped more samples than the previous 40 years put together. And so we've had collaborations with lots of other aspects of the government and outside of government, Big Pharma, Department of Defense, all these other institutes. I won't go through all the acronyms because that's kind of government speak, but just trust me, they're all government organizations. And we published on this in a variety of publications, how we made the library and its methods. how it's used for screening, how we can isolate compounds rapidly from this, and then a whole bioinformatics suite that we're using to look through this data. And I will let you know that the methods and the robots and this equipment has been adopted by many other universities, from folks in Sweden to folks in South Africa, to Brazil, to the University of Mississippi, to University of Michigan, to some corporations have come to get the plans for how we've made these robots and put it into use there too. So now that people are going to get this library, they're going to go screen it, they're going to get a lot of hits, and then they're going to go, what do I do now? I need a natural products chemist. And they're going to call Carrie, and Carrie's going to say, I don't have enough time for all of you people, because it's just too many. And so we had to figure out ways that we could help some of these people with the hits they're going to get. And so we had to automate the isolation and the purification, and also some of the aspects of structural elucidation of these compounds. And when I say structural elucidation, that means what is the exact chemical structure of that compound? And so we did that. I'm not going to go through this in detail, so don't worry, it's not on a test or anything. Essentially, this is just a pilot program where we took about 40 extracts that looked like they had activity against cancer. We put them through our initial purification scheme, and then we put them through our phase 2 purification of the active fractions, and then we put them through a combination of analytical data gathering by mass spec, IR, and NMR, which are 3 different methods to look at molecules. And then by putting the pieces of data together generated by these 3 different techniques, we're able to put together a chemical structure like this. And then when we kind of put this all together, we were able, in 3 hours of instrument time with the pre-fractionation, to create a set of fractions, and then 13 hours total to do the sub-fractionation. And you can see data here, which is essentially, there are 22 sub-fractions for each active fraction. And if the box is green here, it means that was active. It was killing cancer cells. We then were able to repeat that, gather analytical data, and isolate and identify 29 compounds from this, 3 of which were new natural products, and do that with a total of maybe about a month of chemist time, which is far less than would normally be necessary to do that number of projects. And I would say that at this level of purification, about 70 to 75% of the molecules are pure, so we can actually deduce their chemical structures. The other thing is, people screen crude natural products and have for years. And so they say, well, we've already screened the crude. Why do we need to screen these fractions? Aren't we going to just find the same thing that was in the crude? And what I will tell you is that we've done now a lot of screens with a lot of people. Probably a dozen different groups have screened this fraction library. 80% of the time, if you see an active fraction, the crude extract is not active at that same concentration. So what you're seeing is chemistry that was hidden before in that natural product extract because of those nuisance compounds or that cytotoxicity, or because the active compound was in a very small % of the total mass. And by doing this pre-fractionation, we've concentrated those active molecules and allowed us to find things that we would have missed otherwise. So these are just some of the molecules we found in that pilot project. And what I wanted to show you is like the structures, but also, are these drug-like molecules? Because you can find natural products, but are they really going to be similar to drugs? What you see here is we did what's called principal component analysis. So we take a look at the structures of these, the molecular weight, how many sp3 carbons they might have, how lipophilic they are, do they like oil versus water? And we compared them to each other. And what you see here is an overlap. What you see with the gray little balls here are all the approved natural product drugs from the period of 1981 to 2010. And what you see in red are all of the molecules that were purified during our pilot project. And as you can see through the main area here, they largely overlap, indicating that the molecules that we're isolating through this process that is now highly automated, much faster, much cheaper, is isolating drug-like molecules. So that's all good. We're done, right? Well, not quite. There's still a lot of biotherapeutics that are out there and things that people have missed for years. So most of the work in natural products are looking at these small molecules. But especially marine aqueous extracts, have a large amount of biotherapeutics, proteins and peptides that might also be active. For example, you might not know this, but there is an approved drug that was made from a protein that came from Gila monster spit. That's something that's unexpected, but this is used for type 2 diabetes and was approved in 2012. Similarly, excuse me, there is another drug that came from the brain of salmon, calcitonin, that is used for osteoporosis and has been approved and used since 2005. And finally, probably most of you have heard of Botox over the years, which is another foreign protein from a natural product that's injected in probably millions of people a year, including perhaps Sly Stallone here. So what about other marine natural products? Well, you might see this snail somewhere if you were walking in the Philippines and thinks that's a beautiful snail. I am going to pick that up and give it to my significant other. However, I would warn you against doing that because this particular snail is one of a family of cone snails. And these snails are actually known because, like most things out in nature, they have to eat, they have to figure out a way to survive, and they do it by capturing prey, as you see with this poor fish here. What's interesting is that they have a little harpoon that they use to capture fish, and it injects a venom. And that venom actually, because they are not cruel, it actually injects an agent that is a pain reliever that's stronger than opioids and is now an approved drug that's used for chronic opioid resistant pain. So though they are going to eat this fish because they have to, they are going to make sure that it will not be in pain throughout the time. And this drug takes effect within seconds of being delivered. So are there more like that out there? Well, one of the things I'd like to tell you about quickly is something we found here at the NCI, which is a protein from this red algae that came off the coast of New Zealand. It's already been in 2 phase 1 clinical trials for HIV prevention, and it's had no toxicity or immunogenicity. But one thing, I published a paper about 10 years ago showing that this was active against all viruses of the family Conoviridae, so all the coronaviruses. And as you might expect, 10 years later, this became a very timely paper, shall we say, and many more people read it last year than probably had in the previous 10 years put together. And what happened is we then tested this quickly against SARS-CoV-2, and we saw that it was potently active against SARS-CoV-2. What I'm showing here is data on the right is that as low as about 8 nanomolar grafithsin was able to reduce the viral titer in human epi-airway tissues by 100,000-fold, which is kind of a ridiculously large reduction. The reason this works is because it binds to specific things that are on the surface spike glycoprotein. You might have heard about this glycoprotein because it's what all the antibodies are against. But it is covered with sugars, which are shown here in green on the right-hand side. And that's what grafithsin binds to, those sugars. And what is important is that it has 2 binding sites on spike, but it doesn't bind the receptor binding domain, which you see up here at the top in purple and red, where the antibodies bind. Why that is important is because it doesn't overlap with the antibodies, so it can be synergistic with them. But most importantly, all those COVID-19 variants we've been reading about from South Africa, from Brazil, from England, all of those mutations are in this receptor binding domain. And so grafithsin is equivalently active against all of those variants. And so we've tested it already, and it looks like it has similar activity against all of the variants. And so it's going to be hard for the coronavirus to become resistant to grafithsin. Just to show you, we've done animal studies in both mice and hamsters, and in both cases, grafithsin is able, with just sniffing it up the nose of an animal, to block the coronavirus and reduce its viral titers in the lungs and prevent weight loss and symptoms in animals. Another interesting thing about it, here I'm showing data. What you can see in pink is we actually made zircon encrusted grafithsin, which you can see in a PET scanner. And when you look at it, you can see that it is retained up here in the nose. So if you shoot this up the nose of an animal, it stays there 60% at least 24 hours later. There's no systemic absorption of grafithsin. And so what the idea is that you could use this as a drug that would be administered once a day. You could just sniff it up each nostril. It would protect you then for 24 hours. And this could be used to prevent coronavirus from spreading or you from getting it. And should you somehow still get it, it would also significantly reduce your ability to spread it to others. And so for all those folks who, for whatever reason, aren't taking vaccines or can't take vaccines or are immunocompromised, this would be useful. And so currently we have 2 clinical trials that are scheduled, one at the University of Pittsburgh and one at the University of Louisville, that are going to take this into the clinic, and hopefully we'll find out some results that are positive. Another use of it that I show on the bottom is in an inhaler that would actually get it down into the lungs. Because we also found that if you already have the coronavirus and you breathe this into the lungs of animals, it protects the lungs from the damage the coronavirus causes. So if someone came to the doctor and said, oh, I think I've got it, and they have it, but their symptoms aren't too bad, they could get an inhaler like this, which would keep their symptoms from getting worse in their lungs. So with that, I hope I've convinced you to some extent that natural products are a very cool area of research, that they're making an impact today and have for a long time in drug discovery, and that hopefully, I've convinced some of you that this would be a really awesome area to get into. I will let you know that we already have here at the NCI 2 alumni of Oregon State University, one a recent alumni who is a post-baccalaureate researcher, Grant McConaughey working in our laboratory, and Chris Thornburg, who's one of our senior scientists, also an alumni from Oregon State. So hopefully more of you will want to come here and work with us in the future because you're making great contributions already. And thank you very much for your time.
Thank you, Barry, for that exciting and inspiring lecture. It really illustrates the power and importance of a coordinated effort to study the chemical and biological diversity available in nature. And now I would like to invite my OSU colleagues to join me in describing their marine natural products research for our panel discussion and Q&A session, which I think will really highlight that natural products research is such a highly interdisciplinary field. As you've just seen, it encompasses many of the biological, chemical, and computational sciences, as well as engineering. So to determine the possible uses or applications of natural products, scientists from different disciplines frequently collaborate to understand natural product structures, the biological activities, what does the molecule look like in 3 dimensions, what are the genes and enzymes used by the natural product producing organisms? How did that molecular structure evolve? And what is its function in the environment or in laboratory tests of biological activity that really allow the translation of natural products into drugs or everyday products, as Barry's just very aptly described, that even address pollution and climate change, so beyond medicines too. So just about my lab briefly, our overarching goal, as has already been mentioned, is to find and characterize new natural products with useful biological properties. We rely on a premise that is central to natural products discovery, which is that biological diversity equates with chemical diversity. So to find these new natural products from marine environments, my lab has collected cyanobacteria or filamentous blue-green algae in the Red Sea here, and as part of an international cooperative biodiversity groups project in Panama. We've also collected cyanobacteria in the form of biomineralized stromatolites, living rocks from the Eastern Cape of South Africa. The South African East Cape coast has a unique habitat created by the meeting of the cool Atlantic and warm Indian Oceans. And we've also studied tunicates and their microbial symbionts, which are extremely interesting from South Africa. Finally, in collaboration with Dr. Andrew Thurber here at OSU, we currently look at deep sea sediment microbes from methane seeps right off the Oregon-Washington coast by ROV. So we work then closely with Jane Ishmael's lab to choose which natural products to focus on. As you heard, the crude oil that contains the natural products extracted from organisms are extremely complex, and we only want to purify the biologically active pure compound, perhaps that may ultimately become a drug lead or a sunscreen. So once we have worked out the chemical structure of the natural products using the analytical chemistry techniques you also just heard about a little bit, mass spectrometry, nuclear magnetic resonance, or NMR, we give the pure compounds to Jane Ishmael's lab for further investigation of their biological effects. So with that, I would like to hand over to my colleague, Jane Ishmael in pharmacology.
Oh, thank you, Kerry. I'm very pleased to be here. As Kerry said, I am a biologist and more specifically a pharmacologist. So a lot of my work is to try and work out how the mechanism action of molecules. And the teaching part of my job is to teach future pharmacists how drugs that are currently used work in the body. But my real passion is my research that goes on right here in the pharmacy building, and that is I'm one of the first people to look for unusual biological activity with some of these extracts and semi-pure compounds that Dr. McPhail's lab gives me. So I work my students work alongside the natural product chemistry students, and we have a variety of assays using robotics in the pharmacy building. And we're looking for really with a special emphasis on anti-cancer compounds. And we're not just interested in how a compound might kill a cancer cell, we're also interested in how we might stress the cancer cell, and that difference between cell stress in a non-toxic way, so that we might be able to eventually utilize a new drug or discover something that could actually be used in combination with other treatments. And so we use a lot of cell-based models and cell-free models, and we do things on a very small scale, because I've learned in working with natural product chemists that these molecules are very, very precious, very, very tiny amounts. And so we focus on types of cancer that have remained very difficult to treat. So such as glioblastoma, which is a particularly lethal kind of primary brain tumor. And the prognosis for people diagnosed with a glioblastoma has not really changed for about 40 years, really, and the life expectancy, the median age is about 15 months survival. And so we work on glioblastoma and some forms of breast cancer that have remained difficult to treat, such as triple negative breast cancer. So in my lab, we both screen, so we do screening and we work side by side and alongside Kerry McPhail's students and her lab, but also we are very privileged to be one of the first labs to then really get to test the new compound and actually work out how it works. And in collaboration with the NCI and the Drug Discovery Programme, we are also lucky enough to be able to send compounds back to the NCI to see if they profile and match other things that have already been discovered. And so it's almost a little bit like detective work. You're looking for patterns in signaling molecules to give you clues as to how something might be working. We're very, very excited when we find something that doesn't match anything at all that's in Barry O'Keefe's library, because, and I'm very happy to say that we've had a few of those, and one in particular that we're working on now and are funded to study by the NIH. And so we do what we can with small amounts, and we look in many cell-typed assays to preserve the use of animal lives and think about how to conserve and then also conserve the material, because ideally we wouldn't like to keep going back to the original source to collect. We're very aware of that and the ecological impact we could have by looking or keep repeatedly going back to Panama or the Red Sea or South Africa. So we are really delighted if we can attract the attention of, say, a synthetic chemist from around the world. So some molecules are much easier to synthesize in the laboratory than others. And so we've been lucky enough to then be able to collaborate with synthetic chemists and other experts around the world. So it's a very international kind of project, very exciting, but also we're constantly looking for other ways to supply the natural product, because supply is always an issue, being able to take something forward from this preclinical development into something that could be then tested or for broader dissemination to the scientific community. So I'm going to segue now into my colleague, Dr. BJ Philmus, who's going to talk to you a little bit more about how to tackle this problem of supply with a natural product. Thank you.
Thank you, Dr. Ishmael. Hi, I'm BJ Philmus. As mentioned earlier, I am a medicinal chemist, and the research that my lab, so myself and my students do, is focused to kind of bridges natural products, so the chemistry, and also kind of bridges into synthetic biology, where we try and move genes around and clusters of genes around to solve this supply problem. We work a lot with cyanobacteria and cyanobacteria compounds, hence my background of the wonderful green organisms that I work with. And what we try and do is we try and link a molecule, so say some of the molecules that Dr. McPhail and Dr. Ishmael work with and have found, to the producing gene clusters. And then we try and introduce those genes into an easier to grow organism that we can cultivate in the lab to produce it on larger scale, so that we can do more biological assays, more clinical trials, and then hopefully if something is really productive in that fashion, be able to create a system where we can generate enough of this with minimal environmental impacts for actual drug production. And so in addition to that, we also kind of mix and match genes to create compounds that don't actually exist in nature. And we love to study the chemistry of how nature, as Dr. O'Keefe mentioned, nature is probably the best chemist. And I think the one phrase I heard was, synthetic chemists think they're so good, but nature not only first synthesized the molecules, but also synthesized those chemists themselves. And so we're just trying to keep up with nature really and just learn from it. And there are a lot of interesting enzymes that you can use to perform really difficult reactions that are really harsh chemically and require a lot of money and things to dispose of and can be environmentally unfriendly, but nature can do them in water in a cell. And so can you take advantage of that to help in drug discovery or in drug production? So with that, I will pass my time over to Dr. Mahmud, who will talk to you a little bit more about his lab's research.
Hello, my name is Taifo Mahmud. So we have heard a lot about natural products, the very exciting structures with a lot of different biological activities, anti-cancer, antibacterial, antiviral activities, and so on. And my lab, basically, in addition to drug discovery, also we like to understand how those molecules are made in nature. So we study the genes and the enzyme that are involved in making those molecules and use the knowledge to generate analogs of natural products or make new compounds through genetic engineering and synthetic biology, mixings and matching genes from different organisms. So one of the projects that we have related to marine natural products is the related to marine-derived sunscreens. So apparently there are many sunscreens produced by marine organisms, from dinoflagellate, diatoms, cyanobacteria, like the picture behind Dr. Philmus' screen, and also marine invertebrate. And you can also find sunscreen compounds in like the eyes of mantis shrimp or in fish. But in the case of fish, it has been believed that those sunscreen compounds are obtained from their diets. But several years ago, we found through our genome mining studies that actually fish can produce their own sunscreen compounds. So apparently fish can obtain sunscreen from the environment, from their diet, but also they can produce them. So there's two different types of sunscreen. One protect them from UVA and the other one protect them from UVB. As you know, marine environment is very harsh in terms of UV irradiations. And of course, humans like us, we also need to put sunscreen on us. If you go to the ocean, to the beach, then you would put a lot of sunscreens. But the problem is some of the sunscreen product, or most of the sunscreen product, contain synthetic sunscreen compounds that have negative impact, environmental impact to the ocean. So many of them can cause coral bleaching or kill the larvae of corals. So definitely we need to develop new and natural derived sunscreen products. So for that reason, we studied the biosynthesis, or how those compounds are made. And then we use the knowledge using biotechnology approach. So what we did, basically, we took genes from fish and put into yeast, and then produce sunscreen by fermentation. And that way, basically, we can produce sunscreen product without damaging the environment. So there's technology that was developed here at Oregon State University, now being licensed by a biotech company here, also in town in Corvallis, called Gadusol Laboratories. So yeah, with that, I think I will let Carrie to take over this again.
Thank you, Taifo, and thank you, Jane, BJ, and Taifo for the insightful descriptions of your research. It's really just an incredible pipeline that we have at OSU. So we'll now open up the Q&A session moderated by Hayleigh to address as many questions I think as we can get to by our 5 p.m. end time. So with that, I invite Hayleigh to join us.
What sort of instruments are commonly used for the pre-fractionation process? Because you mentioned that they can clog up the expensive ones.
Right. So when people are doing high throughput screening, they have instruments that can collect and take liquid samples from a plate, often 384 wells in a little plate about the size of, not a bigger phone like that. And so if they have very fine needles on those to take up that liquid. And if you have crude natural products, they can be viscous, there can be all sorts of things in there, and they can be difficult for them to pipette in. So that's a challenge. What we do with the pre-fractionation is we separate pure molecules out of there by doing a step of chromatography. In this case, we put a media in a column, we put the extracts on there. Ours is actually a two-stage system that puts the extract on cotton first and then puts it through a particular type of media, chromatography media. And then with that robot that you saw moving, we push through different solvent systems, and they have increasing or decreasing amounts of water versus methanol or acetonitrile. And that separates the compounds based upon their polarity, which is basically how much they like oil versus water. And so you end up with some compounds that are concentrated in those individual fractions. Those are then dried down and put on those plates for the screeners to use, and they are much better behaved than the crude extracts. And so we use robots, we use lyophilizers, we use chromatography media, we use liquid handling robotic systems. And so there are multiple different types of systems. And it's a lot of automation, but one individual with a pipetter having to do something 1,000 times is not good for that individual.
Thank you. And the next one is slightly related. You mentioned that a lot of the instruments are 3D printed. Do you happen to know if those plans are publicly available?
We are making them publicly available. So we've had requests from that. Right now, we are actually had to buy some new software to set up automated displays of how these components work with the robotic systems and how the robotic systems, because we've been explaining them to group to group to group, and it's challenging. So we're right now writing software, getting the programs together. We already have the CAD designs for all of the components that we made that go with the programs. We'll probably publish a paper overall on the automation in general and make all of that information publicly available.
Sounds fun. In the microbes that are used as natural products, what proportion of those are bacteria versus fungi? And do you happen to be sequencing genomes for publishing or public access?
You mean here at the NCI? I was going to say BJ probably knows more about what specific things are fungi versus bacteria. As far as us making things public, there's a balance we have at the NCI, because what countries allow the NCI to collect organisms there, whether they're microbes or marine organisms or plants. And those countries, as we call them, host countries, have certain benefit sharing rights that are important. So it's a balance between the rights of those countries to receive some benefits should something come or be commercially developed from a source organism from that country, and the benefit of making that information broadly available to everybody. And so what we try and work out is mechanisms where that information can be made available to people, excuse me, but that it has to be done with an agreement that should they come up with something based upon their access to that information that becomes a commercial thing, that they will share benefits with representatives of that host country, so that that country, whether it's Indonesia or whether it's Peru or Ecuador or Kazakhstan, will receive some benefits for allowing their biome to be investigated by researchers. And then BJ, I'll leave it to you whether there's more stuff in fungi or actinomycetes or cyanobacteria. I think there might be a variety of opinions.
I think according to your, well, now former colleague, Dr. Dave Newman, macroorganisms don't make anything. It's all the microbes that they host. And you know, for that, then I say bacteria wins out because more of them are hosted than fungi. So that's my personal, not in any way biased opinion by the fact that I grew up with bacteria. So that's me.
Can I jump in? I do think that's true. You know, we started off looking at bacteria, right? There were more people, I think, researching bacteria, and they're a little bit more accessible, easier to work with in some ways, given that they're prokaryotic. So maybe that balance is going to change. And I did see also that somebody had asked about whether you know, the field of natural products research is going to move more towards engineering. And I think that BJ and Taifo can both answer that very well.
Go ahead, BJ.
Yeah.
So do I think it will be dominated by engineers? No. Do I think that they have an incredibly important role to play? Absolutely. And I think that the more interdisciplinary between science and engineering and getting engineers into labs to just see what the problems are, and having students and faculty and postdoctoral researchers and staff scientists going, this is enough. And how do I phrase this? The best scientific advances seem to come out when someone just gets incredibly frustrated with the way they have to do something and they go, I'm done with this. And then they go find a better way to do something. So just the more mingling you can get with engineers, I think, I think is great, because engineers just have a way of conceptualizing and building things that sometimes scientists don't, because we're sometimes more like, oh, I'm really interested in the molecule. And they're like, nah, you don't really need the molecule. You need a better way to do all of this. And so wasn't quite mentioned, but part of one of the little programs we're starting up at OSU in collaboration with the College of Engineering is we're trying to get particularly undergrads, but senior undergrads that have capstone projects in engineering, and having them meet some of the graduate students and undergraduate researchers in labs and trying to solve problems. One of the simple problems we solved this term was one of my students grows cyanobacteria and they use glass tubes, and obviously we don't want to throw them out because that's a huge waste, so we wash them and rinse them. They're pretty much doing that all by hand. And so if you do an experiment that has 96 tubes, that's, you know, not as much fun to wash as you might think. And so the engineering students came up with a great simple design to just automate the washing and the rinsing. It's, you know, it's not, it was nothing like, you know, they didn't have to weld anything. It's all made out of, like, you know, plastic and drilled holes and stuff you can buy off a plumbing shelf, but they engineered it and they tested it, and they like, okay, it doesn't work right, so they got it to work better. And so I think things like that are hugely important and beneficial to lab workers.
If I can add to it, you know, dealing with natural products definitely require a deep understanding of chemistry and biology. Just engineering itself is not good enough, or will not be successful in getting a new natural product, or even for separation or isolation of natural products. So definitely somebody like Dr. Barry O'Keefe, you know, who has a very good background in biology and chemistry, and also interested in you know, modernize the technique and use the engineering approach to improve the outputs. And I think moving forward, still, you know, basic knowledge of chemistry and biology are still important.
I agree. If I can jump in quickly, just in general in science for everybody out there, you know, the more you can get of disparate opinions, whether it's your, like Jane, a biology collaborator, whether it's an engineer or someone to come in and join your group, I think the better a lab you have. There's been actually sociological studies done on what are successful labs, and successful labs aren't ones whose first time through an experiment, they have a higher success rate than another lab. All of our labs fail about half the time because we're doing new things, and so you don't know. The labs that are successful are those that have a variety of opinions and outlooks, whether it's an engineer, a biologist, a chemist, who look at things from different angles. And so they get your group out of its groupthink. And you have someone there to say, well, why do you do things that way? Because they didn't grow up through the same systems. And that raises the question, which makes you think about it, well, why did we do it that way? And that more quickly allows you to turn an experiment that didn't turn out right into a new discovery about what the reason it didn't turn out right is and what that's saying about the biology and chemistry. And so I think that by having that mixture of people in a laboratory, diversity of opinion and ideas and backgrounds, that's huge help. And I will say in our lab, we started off with one little 3D printer. And when the chemists and biologists started saying, what, you can make something that makes life easier, suddenly the guy who used to be a chemist and then just learned CAD design and was printing things now has 3 of these printers going constantly, and everybody keeps giving him so much work to make new things, to make everybody's life a little bit easier or to let them do things they couldn't do before, that he's bemoaning the loss of time of being able to do chemistry. So I think once you bring some of these ideas in, everybody starts sharing ideas, and the whole thing is kind of a virtuous cycle.
Great discussion. Wow. So Hayleigh, I think, do you want to move on to some more of the questions? I see the list has been growing.
Yes. Would each of you mind talking briefly about your journey to where you are now and how you got into it? Also mentioning how students such as myself could get involved as undergrads or graduates.
Jane, I wonder if you want to start.
Yeah, actually, that's a great point, Hayleigh, because I trained in the UK first, and I did have an undergraduate research project, and I actually studied mouse behavior. So I started off really as a neuropharmacologist, and the course that I had went through in the UK was called a sandwich course, which means basically for 1 year you go out and work in industry. And I think I worked in Scotland, the pharmacology group there, and then I realised that experience made me decide I wanted to go and get a PhD when I was carry on with the research. That really inspired me into research, and then I ended up working really at the interface of toxicology and pharmacology, and then I think I didn't really imagine myself in academia, but it kind of just sort of happened, really. My inspiration to start to really work in the cancer area also came from a personal loss of a relative in 2006 to cancer, and I was always kind of scared of cancer biology before, but I think it's really along the way a mixture of seeing something that needs to be done, and the idea of discovery. Also realized I really love teaching, and so that's how I ended up at Oregon State. And then also realizing there were some amazing molecules being discovered here, and just the chance to collaborate, really. So my journey was more of a deciding what I didn't really have a really, didn't sort of wake up one morning and say, that's what I want to do. It was more just the people I've met along the way and the experiences, and then just being in a situation to work in a group and do team science, really. That's what it is. And so, and be around other people with other thoughts, just as a kind of a theme that we've been talking about today. Yeah. Thank you.
Thanks, Jane. And Taifo, I wonder if you want to.
Sure. So yeah, so I got my education or training in Japan as a natural product chemist. So I'm a natural product chemist by training, and so my first project was plant natural product. medicinal plants. And then I worked on marine spawns and then synthesis of natural products. And then when I did my postdocs at the University of Washington, that the first time I worked on microbiology and bacterial natural products to study biosynthesis and then to understand how compounds are made in organisms. So I came to OSU in 2003 because OSU has been, as perhaps even today, has very strong core of natural products investigators here. So I was very excited to join OSU at that time. And then, time flies, it has been 18 years now. And so many of my projects really related to natural product biosynthesis and discovery. And to tell you the truth, many of them, many of new direction really occur serendipitously. Basically, you didn't plan of doing it but based on what we have done, and finally we have a new direction of research. So that's make research in natural products is very exciting because you never know what comes. For example, when we do genome mining study, just looking for a genes that is involved in making anti-diabetic drugs, we found the same genes also in animals. So we were surprised because usually that gene's always in microbes. That's the reason why we look for it, and we found fish can use that genes to make sunscreen compound, for example. So that's I think the reason why we survive with all the difficulties and challenges in research in natural products. Thank you.
So true. And Dr. Philmus, BJ.
So how did I get here? When I was an undergrad, I started, I was a marine biology major, and I had this, I was going to be a dolphin trainer, and and then I realized I didn't want to stand up in front of large crowds in a wetsuit all day. And then I ended up taking a course in chemical ecology, and it really opened my eyes as to how much the chemistry really influenced all the biology. And if you really wanted to think about the biology, you had to understand the chemistry and the molecules that were being traded back and forth between all the organisms. And that just kind of got me into natural products chemistry, just by being excited in that way. And then I ended up doing my PhD at the University of Hawaii, where my PI, we grew cyanobacteria, and I just loved all the different green, and sometimes red and brown and purple. And that's pretty much what I've gotten into. And then I kind of drifted back to chemistry to learn how the biological chemistry worked. And I've always loved taking things apart and putting things back together again. And so now I just do that with genes. And I take genes apart and I put them back together.
Oh, fantastic. Thanks. So I can say maybe my path is pretty similar to some of the thoughts there, that really doing research as an undergraduate was an opportunity that I didn't realize how precious it was at the time. And I was at university in South Africa. I'm from Zimbabwe, and so I had the opportunity to go to university in South Africa, and just very serendipitously was funded to do a PhD. And that was, because perhaps I had this undergraduate research experience and made the connection with people that I loved to scuba dive. I was standing around a barbecue one evening at the dive club, and somebody started talking about natural products chemistry, and I was busy studying marine biology. So that was really it for me. And after my PhD in chemistry, I came to a conference here, the Gordon Research Conference in Marine Natural Products, and met my postdoc advisor, who was from Oregon State University, and actually the rest is history. So as BJ says, it's really thinking about we don't see the communication between animals and plants, and we're kind of blind to this world that, well, now we can see it using mass spectrometry and NMR, but chemistry is the way that other organisms communicate, and they're very sophisticated chemists, whereas it's been said twice that we're not nearly as sophisticated as the microbes that make many of these natural products. So with that, I'll pass it back to Hayleigh to carry on with the questions. Thanks, Hayleigh.
Carrying on, this one's a two-parter. Have there been any discoveries regarding sea urchins? And have there been discoveries regarding bone grafting or regeneration of stem cells that assist in a natural scaffold?
Perhaps Dr. O'Keefe can take that one.
Oh, really? You're throwing the urchins at me, huh? Okay.
Or the stem cells, if you want.
Yeah, we certainly have echinoderms in the natural products collection here. In general, I don't think we have isolated anything that has progressed towards as far as preclinical development and doing animal studies or things from them. From either the venoms. We don't collect the venoms necessarily here of things, so that would be other researchers, and I'm not as aware if they have aspects of the venoms that have been investigated and utilized. But certainly, I think with the current overpopulation of sea urchins on the California coast, I don't know if it's as bad up in Oregon as it is in other places, but certainly I think finding a use for them for a medicinal agent or a sunscreen or something else, right now there certainly is an overabundance of them causing some ecological damage. So being able to find something from them, I think is a very good thing to look at.
Yeah, and I must say, I'm not sure, Jane, if you have anything to say about the stem cell research, but I don't know anything about that.
Oh, only that I think really there's through collaboration of people that are studying, I know about cancer stem cells, and that this is always in terms of cancer and whether the natural products actually do anything to the stem cells. We are actively working in that area. That's always a question of interest, and that's probably the easiest thing to say. But no, that field is evolving really. As we know more about stem cells, we learn about the signaling that underlies them, and then we know if there are points to target the stem cells as well as just the other kinds of cells that are in the tumor mass. And so.
Next is, how do you make sure the process of extracting natural products is sustainable?
So maybe I can say a little bit about that as far as from our perspective. Dr. O'Keefe spoke about the Convention on Biological Diversity. It's very important that wherever you get that natural product material from the source organism, that it's documented and that there's an agreement that it is harvested. For the actual collection, I know, for example, the sea squirts or tunicates that we take, we're particularly interested in encrusting tunicates, and we're very careful to take only a part of the colony, a small part of the colony. And, this is really possible now because of the really advanced technology that we have. And that is just such an important thing. By way, what immediately comes to mind is something that I was talking about the other day. We have taken a small core of living rock, a stromatolite, and from that extracted 100 micrograms of a compound, and we completely worked out the structure from that 100 micrograms. And that just was not possible 10 years ago, maybe not even 5 years ago. And this is just truly because of this incredible advance in mass spectrometry. I saw one of the questions was, do we use QTOF, MSMS? Absolutely, every day, every single one of the medicinal chemistry faculty here. NMR, nuclear magnetic resonance spectroscopy as well. We can put the sample in a very, very small tube, like 1.7 millimeter diameter. And at OSU, we've got 3 millimeter or 5 millimeter probes to put the tubes in. But we also have very high field instruments, and these very high field instruments bring much greater sensitivity and resolution. So I think that's the best way to ensure sustainability, is that, honestly, from the stromatolite sample, we can't go back to the environment. South Africa is a very long way away. I have collaborators there, but we cannot cut enough rock out and extract it to get enough of this compound. So already, as Jane Ishmael actually alluded to, we've passed the structure onto synthetic chemists. We also work with people to do metagenomic gene sequencing to mine the genome and look for the biosynthetic gene clusters, as Dr. Philmus has talked about. So already we think we have the genes for that biosynthetic cluster. And so whether it's chemical synthesis in a lab or biological synthesis by expressing the genes in an organism, even synthetic biology, we'll get the source from somewhere that is not the original organism in the ocean. And that will be enough for testing to see whether it's good enough. And I think that that's really important, because many of the compounds don't go anywhere yet, or they don't go anywhere for a long time. So there's no reason to collect, a large amount of them. And so, Dr. Barry, I'm not sure if you've got anything else to add.
No, I mean, I would agree completely. I think the single biggest thing you can do, and the biggest advances we have are just what you were talking about, the technologies. It used to be, I started in the natural products branch maybe 5 years ago. At that time, they used to send out 10 grams of an extract for someone to work on. And we couldn't take it back because, we don't know how it's been used or things like that. So that was gone. Now with what we're doing with pre-fractionation and sending the samples out, we can do the same amount of work on about 75 to 80% of the things using 250 milligrams of the crude extract and 1 milligram or less of the fraction that's derived from that, and then get the same amount of data from that. So simply by doing that and reducing the amount of usage, you could preserve, in this case, a library of hundreds of thousands of extracts for, double, triple, quadruple, sometimes even 10 times how long they would have lasted as research resources previously. And that's due to the ability to do this with very small amounts, identify the structures, and that we can screen in very tiny amounts. When we ship samples out to people, they get 10 micrograms to screen, and that's enough to do multiple screens. And so by conserving what we already have, that's the first step in being able to make sure that we aren't decimating any type of natural resource or ecological niche that's out there.
Okay, can I add, it just strikes me, it reminds me that one of the students in Jane's lab did his entire PhD on 0.2 milligrams, so 200 micrograms of material, of pure compound that we gave, all 5 years of PhD. And I was just stunned the other day that 100 micrograms of one of our recent compounds would be enough for the pilot mouse studies to test it in preclinical setting. Just amazing, because these compounds are, very potent at what they do, very good at what they do.
Yeah, the next one is for Taifo. Do you know of any naturally derived sunscreens on the market now?
Well, if zinc oxide and titanium dioxide are considered as naturally derived, yes. But those are minerals. And so far, there are no pure natural compounds being used, but there are, extract derived from red algae that contains sunscreen compound that I mentioned just now. So like dulse and other red algae like nori in Japan, those contain sunscreen compounds. And some cosmetic companies in Europe, like in France and in Switzerland, they produce extract of red algae, and then they incorporate that into their cosmetic products. So it a little bit sounds like natural sunscreen, but the problem with using extract, of course, you don't know how much the compound is in there, and then depending on when they harvest the algae, the concentration of the compound could be different. So of course, this is just an addition, as an addition to the cosmetic recipe that they already have, containing zinc oxide or titanium dioxide. But real pure natural product has not been used in sunscreen products.
Thank you. Someone noted that red algae compound can be grown in tobacco plants. Are there other natural means of synthesizing otherwise difficult to collect marine compounds or other sources?
Yeah, so there are several natural products being produced in a different host. And then what we are doing, and Dr. Philmus doing as well, basically, taking genes from different organism that the original producer, but maybe they are not easily obtained, or maybe they are endangered species. Then you can just take the genes and then move into a more manageable host, or like bacteria or yeast or something like that. Even tobacco plants is being used as well. So there are some, well, actually, like the anti-malarial drugs, for example, being expressed in yeast and E. coli. So yeah, there are examples. Thank you.
All right, this one is for BJ specifically. Why can't cyanobacteria or other organisms that make these bioactive compounds just be cultured?
Yeah. So that's a loaded question. It's not that, so some of them just don't grow in culture. Like in a lab, they don't grow. They either need a specific nutrient from the ocean, or one of their, so I don't have a picture of, maybe that should be my background now. They have a lot of, so the cyanobacteria have a lot of other bacteria that grow on their surface. So they carry a lot of hitchhikers with them. So it's possible that one of those dies, and then the signaling between the bacteria and the cyanobacteria, then that stops, and then they stop producing a compound. So a lot of things. In a lot of things, it's a lot of them from nature, and I won't say that this is general, but it feels like it. The more interesting the compound that anything produces, usually the slower growing it is. And so it's not that they can't be cultured, it's that you can't culture them fast enough to really get enough of the compound on the kind of time scale that you really need to supply the amount you would need for practical use. So that's the short, long-winded answer.
If I can, I'm going to make a quick plug here for biotherapeutics, potential here, because, nature has a lot of different ways to evolve, ways to survive. So as BJ mentioned, something that grows slowly probably needs to have more chemical defense than something that grows really quick and can keep growing faster and recover from damage from predators or other things in the environment. But similarly, proteins and peptides require a much less of a genome that an organism has to carry around with it to make it. If you have, like streptomycin, you might need 27 different enzymes to make that small molecule. But something like grafitsin that I talked about, that's like maybe 600 base pairs practically. It's a very small amount of genetic material that must be maintained. And so that allows some efficiencies evolutionarily about kind of how much baggage you've got to carry with you to be able to fight off whatever you were trying to fight off in your environment. So I think there is a lot of room in the area of biotherapeutics, and I think they have some advantages because transferring those genes and getting expression of those peptides and proteins in heterologous hosts is in many ways a little bit easier than some of the significant challenges BJ and Taifo take on, which I certainly would not take on myself. So just a quick pitch for them.
Very. I have a question. How did you get Griffithsin enough for your clinical study?
Well, that was kind of a walk through evolution. We took the gene, originally we isolated the amino acid sequence. We made a synthetic gene. We put that gene, showed it could be expressed in E. coli and that it was okay. We then took that plasmid and transferred that into tobacco mosaic virus. So now we're in a virus and put it into there. Then that tobacco mosaic virus was used to infect tobacco plants, didn't create a true, transgene. It just infected the plants, produced a lot of Griffithsin, and then 12 days later you harvest them. So it's not a true genetically engineered plant. And then that plant produced gram quantities of Griffithsin. And so each tobacco plant produced almost a gram of 99.8% pure Griffithsin with just a 3-step purification. And that's the material that's being used for the clinical trials now. We are looking at significantly larger scales because things might be moving in the clinic. And so you can make, multiple gram quantities in E. coli per liter of E. coli growth. Yeast, they've been looking at a variety of other systems to express it as well. And so I think, like with BJ and other, it's a part of finding a good host that is going to be able to express and tolerate the expression of these foreign proteins.
Thank you.
Thanks.
Next is for Professor Ishmael. For the brain cancer cell lines, how are you looking at the issues associated with getting complex natural products across the blood-brain barrier?
Yeah, that's a great question. And I think the best way to think of it is that you have to be really patient. And my role in this discovery project is really at the very beginning. So I start off with not worrying about the blood-brain barrier issues and first try to use these molecules as probes to understand why these cancer cells don't want to die. And really what you, by first using them as probes to understand biology, that's a baby step. And very often in natural products drug discovery, the final drug entity is often not actually the original natural product. And so along the way, we will generate additional tools, so sometimes a simplified molecule, which then, or understand a new target, which then gives people a fresh idea or another way to look at it, and maybe to step this preclinical study further on. And so that's one way to think of it. Sometimes we're just looking at the biology of a glioblastoma cell and what might be a new target that could be accessed then in a different way. The other issue with brain tumors that frequently is the blood-brain barrier is actually compromised and leaky. And so in terms of, so you're exactly right, there are very few chemotherapeutics that actually get across the blood-brain barrier. And this is a general drug delivery problem, which is requiring bioengineering and new kind of formulation and new ways to look at it. So it's a bit like that game of telephone. I'm right at the very beginning, but if we keep going, eventually we hope it'll reveal new ideas, new druggable targets and new molecules to actually help these particular group of patients. That was a great question. Thank you.
So I see we're getting close to 5 o'clock here, or 8 o'clock on the East Coast. And there was also a question I noticed that I just think has really been illustrated by the discussion and the talks today. The question is, how important is the continued collection of marine samples to develop drug products? And I would say, it's absolutely critical. When you hear about also the compounds that people have repurposed towards the pandemic right now, the COVID-19 crisis, some of those compounds like Aplidin have been, are natural product derived. There's many other antivirals that are natural product derived and being able to pivot so quickly was because there was this library available, that there were these libraries available of natural product compounds. So to me, that's just a brilliant question, because I think the answer is it's absolutely the most important thing that we can do is to describe the chemical diversity and the biological diversity that we have access to for all sorts of everyday products and also human health treatments. So with that, I think I'd like to invite Jack to come back and maybe close for us.
Thank you, Carrie. I was watching all those questions go by. That was really amazing. Thanks for the discussion. So I'll just thank again our feature speaker, Barry, and Carrie for organizing, and of course our panelists, Jane and Taifo and BJ. And Hayleigh, great job getting through all those questions. Thanks for being here. And then finally to Oshi Media Services and above all, Virginia Nayland for getting this all together. So really exciting. My brain is spinning. I've been on the web looking at what Griffithsin is, and that's pretty cool. Thanks again, and hope this motivates us to keep talking with each other. Have a good evening.